name's Elijah, my partner's name's Jayden, and this is our DNA replication stop motion. DNA replication happens in the synthesis phase, part of the interphase in the eukaryotic cell cycle. It happens to ensure that all cells that divide through mitosis are genetically identical.
This is a diagram of a nucleotide, which is the monomer of a nucleic acid. Nucleotides include a pentyl sugar, deoxyribose for DNA or ribose for RNA, a phosphate group on the left, and a nitrogenous base, which can be adenine, thymine, cytosine, or guanine, or uracil for RNA. The numbers in the diagram represent each carbon of the sugar, and the arrows indicate the direction you count the numbers, which is clockwise, starting at the oxygen atom. These numbers will tell us whether the DNA strand runs 3' to 5' or 5' to 3'.
There are four main enzymes used in the DNA replication process. Helicase, the cutter, which will be played by this amazing red blob of Play-Doh. DNA polymerase, the builder, which will be played by this equally amazing blue piece of Play-Doh. Primase, the starter upper. And in yellow, ligase, the gluer.
This is the enzyme helicase. Helicase cuts the DNA strand in half by breaking the hydrogen bonds connecting the nitrogenous bases of one strand to the nitrogenous bases of the other strand. This process allows the new strand to be semi-conservative because half of the new strand is from the original strand of DNA.
Now that the DNA strand has been cut in half, it is primed for the primase enzyme. Before DNA polymerase can start building new sequences, it needs something to build off of. It can't actually start a new strand itself. It has to add on to something. This is where DNA primase comes in. It synthesizes or builds the beginnings of a new DNA strand with the correct bases in the correct spots with reference to the parent strand to start things up. It does this in the 3' to 5' direction going off the parent strand's labels. But because complementary DNA strands are anti-parallel to each other, it starts at the 5' end of the new DNA strand it's currently making and heads towards the 3' end of the new DNA strand it's currently making. The reason that DNA primase can't just build the entire strand is because it technically builds RNA molecules using ribose instead of deoxyribose and uracil instead of thymine. DNA primase is a type of RNA polymerase.
DNA polymerase is the third enzyme used in DNA synthesis. DNA polymerase builds the new DNA strand starting from the primers. DNA polymerase adds nucleotides so that the base pairs match. For example, if the original strand contains adenine, DNA polymerase would add a nucleotide with a thymine as its base. Adenine always pairs with thymine, and cytosine always pairs with guanine. DNA polymerase can only move in one direction, which is 3' to 5' on the parent strand. Because of this, DNA polymerase adds DNA from 5' to 3' on a newly formed strand. Because DNA polymerase only moves in one direction, the DNA polymerase following the helicase can continue to move freely without stopping. The strand this DNA polymerase is on is called the leading strand. On the other hand, the strand with DNA polymerase going away from the helicase is called the lagging strand. Since DNA can only move from 3' to 5', this DNA polymerase has to build the new DNA strand in chunks called Okazaki fragments. Because the DNA polymerases on the lagging strand will have to jump back to helicase, many DNA polymerases work together on the lagging strand to hasten the process. Finally, after completing the new DNA strand, DNA polymerase goes back to the DNA primer and changes the RNA segment into DNA.
However, the DNA replication process isn't done quite yet. It's not uncommon for a few breaks or disconnections in the created DNA strand to occur. Ligase, often referred to as the gluer, reattaches the sugars of any broken nucleotides with something called a phosphodiester bond. I think that's how you say it. I googled it.
In the end, the original DNA strand replicates itself, creating a total of 2 strands. So at the beginning of the S phase, each chromosome is made up of one chromatid, but after the S phase, each chromosome now has 2 sister chromatids. These are sister chromatids because they have identical genes and are connected by a centromere.
Thank you for watching our DNA replication stop motion. This may be the end of the video, but it's never the end.